Welcome to part 2 of this course about the Outbreak Investigation tool. The first part provided an overview of the tool, and this second part gives some more practical information about conducting outbreak investigations of PPR-like disease. To get the most out of this presentation, it's important that you've read Section 6 and Annexes 9, 10 and 11 of the ECo-PPR Field Researcher Manual. So if you've not yet done this, pause the video and do it now. The aim of investigating PPR outbreaks is to determine whether livestock keepers' reports of PPR-like disease are truly caused by PPR virus. PPR disease has a range of clinical signs, and the signs can vary between outbreaks. Some flocks show a mixture of signs, while others show mainly respiratory disease, and some show mainly diarrhea. Some outbreaks are very severe, with a lot of animals affected and high mortality, while other outbreaks can be mild with few deaths. PPR signs overlap with the clinical signs of other respiratory and intestinal diseases. This means that sometimes other diseases are wrongly diagnosed as PPR, and sometimes PPR outbreaks are wrongly diagnosed as other diseases. In this project, we would like to use the PPR Rapid Diagnostic Test and laboratory tests to confirm the PPR diagnosis in as many outbreaks as we can, to get a better idea of the true incidence of PPR in the study area, and to get a better idea of the impact of PPR in the area. Any positive samples that you collect will also be sequenced, and phylogenetic analysis will help to show how PPR viruses are circulating and spreading. We will link these findings to our studies of flock movements and trade networks. It's important to prepare in advance for outbreak investigations so that you have all the equipment ready whenever you receive a report. You need to be prepared for interviews, clinical examinations, and sample collection. The investigation team should include a vet who can do the clinical examinations and collect samples, and someone who speaks the local language for good communication with the livestock keepers. When planning the visit, it's important to make sure that the flock will be available and hasn't left for grazing or watering. The first step is to interview the livestock keeper to get as much information as possible. Refer to the outbreak investigation form, which has a list of questions to ask. It's important to get an estimate of the total number of animals in the flock, if possible by species and by age, so that we can calculate the proportion affected and the mortality rate. In many places, it's not culturally appropriate to ask how many animals there are in the flock, but you can roughly estimate the flock size by looking at the flock yourself. The local language name that the livestock keeper uses for the disease problem is a useful piece of information. Always write this down, and also try to find out the literal meaning of the name. Now let's take some time to get familiar with the outbreak investigation form. Go through the form in detail, and if you have any questions, make a note of them and raise them during the live training sessions. The next step is to have a look at the flock. Look at it from a distance and estimate the approximate size by species and by age group. Get a general impression of the main clinical signs affecting the flock. Also, listen out for abnormal sounds like coughing and sneezing. The next step is to select a few animals for clinical examination. Choose animals that represent the range of signs that you see in the flock as a whole. Be systematic with your clinical examination, starting at the front of the animal and move backwards to the hindquarters, paying special attention to the eyes, nose, mouth and lymph nodes. Observe how the animal is breathing and check for evidence of diarrhea. Finally, take the rectal temperature. When you examine the mouth, make a note of the number of pairs of permanent incisor teeth, which is a good indicator of the animal's age. See more details of how to age by dentition in Annex 11. Based on the interview with the livestock keeper and your clinical findings, you can now decide whether the disease problem fits the case definition for PPR-like disease. If the disease problem fits the case definition, then you should run some PPR virus rapid diagnostic tests. Choose up to 5 animals with early clinical signs of PPR, because these are the ones that are excreting most virus. These are animals that have been sick for only 1 to 3 days and have a fever and eye or nose discharge. They may also have other signs, but it's important that they've not been sick for a week or more, because by that time they'll be excreting very little virus. Follow the kit instructions exactly, using a nasal swab from each animal. Test one animal at a time. As soon as you get one positive test, you can stop. One positive test means that the flock is infected with PPR virus. Samples should also be collected for submission to the laboratory for PCR and sequencing of positive samples. You should collect samples whether or not you get positive rapid test results. If the rapid test is positive, then the lab samples will be very valuable for virus sequencing. If the rapid tests are all negative, it's possible that the outbreak is still caused by PPR virus, but the animals are not excreting much virus. We therefore send the samples to the lab for PCR, which is a much more sensitive test than the rapid test. From each animal, you should collect eye, nose and mouth swabs, which are put into cryovials and put in the cool box or liquid nitrogen. You should also collect a plain blood sample, which should be kept cool and the serum removed later and frozen. More details of the samples to be collected and their handling and storage are given in Table 1 of Section 6 of the manual. The final part of the investigation is to carry out post-mortem examinations on any goats or sheep that have recently died, preferably within the last few hours, but up to 24 hours can still be useful. As for the clinical examination, be systematic. Examine the carcass from nose to tail, then cut open and examine the chest cavity. Look at the lungs, the heart, the lymph nodes, and then the abdominal cavity, especially the small and large intestines and the mesenteric lymph nodes. Collect tissue samples from lungs, lymph nodes and spleen for laboratory submission. Finally, having found a positive flock, the next step is to determine the extent of the outbreak. If other areas are identified as possibly affected, then PDS and/or outbreak investigation should be carried out in these areas. Here are some links to further information about the project. If you have any questions about any of the steps of the outbreak investigation, keep a note of them and remember to ask them during the live training sessions. Thank you for watching this presentation, and I hope you found it useful.